Hi everyone, hope everyone is fit, healthy and fine. Welcome to Shine. Today we'll be learning about a new topic, that is cell biology. So this is part 1. In this part 1 we'll be discussing about the structures and functions related to the cell. So what is cell? Cell is the smallest unit, or we can say the structural and the functional unit of any living organism. And the study which is done regarding the cell is called as the cell biology or the cellular biology. So let's see the structures and the functions. So in the structure or the components of the cell, we'll be mainly discussing about the organelles and the cell membranes. So the organelles are being surrounded by the cell membrane, and the functions of the cell is being compartmentalized into the different organelles like nucleus, rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, mitochondria, ribosomes, lysosomes, peroxisomes, and the cytoskeletons. Now the cytoskeletons have three parts, that is microfilament, intermediate filament, and the microtubules. Now let's discuss about each organelles one by one. So the first one we have is the nucleus. This nucleus is also called as the information center, since this nucleus is situated in the center part of the cell. This nucleus is called as the information center because the cell database, that is DNA, is been cell database is been getting encoded, or we can say it is been getting replicated in this region only. The DNA replication is taking place. So its function can be discussed by seeing the structure of the nucleus. As you can see, it is having a nuclear envelope which is protecting or dividing it from separating it from the outer environment of the cell. So this nuclear envelope is mainly providing the strength to the nucleus. This nuclear envelope is divided into two parts, that is the outer layer and the inner layer by an inner membrane space. So on the outer layer you can see there is a presence of the ribosomes, and in the inner layer there is a meshwork of the proteins called as the lamins. Now here there is a presence of the nuclear pore which is acting as a transporter, means it is responsible for transporting the materials from the cytoplasm to the nucleus and from nucleus to the cytoplasm. It's mainly transporting the ions, proteins and the nucleotides. Now here you can see the presence of the nucleolus. So this is the reason where the synthesis or the production of the ribosomes is taking place. Means the these ribosomes which are responsible for the production of the proteins are being getting synthesized in this region. Now here you can see there is a presence of the chromatin. So this chromatin is nothing but a complex of the DNA and a special protein called as the histone. Now this chromatin is of two types, that is euchromatin and the heterochromatin. The euchromatins are the loose ones and they are called as the active region. Now why they are called as the active region of the portion? Because here the transcription process, means the replication of the DNA, is taking place. Now the heterochromatin region is called as the inactive one because here no transcription of the DNA is taking place, and it is tight in nature and it is found near the inner membrane of the nuclear envelope, means somewhat here you will be finding this portion. Now the next the next organelle we have is rough endoplasmic reticulum. Now the rough endoplasmic reticulum is the part of the endoplasmic reticulum networks. So this is mainly responsible for the synthesis of the proteins. Now it is called as the rough endoplasmic reticulum because it has the rough appearance due to the presence of the ribosomes on the outer membrane of the rough endoplasmic reticulum. Now what this ribosomes does, they help in the production or the synthesis of the proteins. Now the proteins which are being synthesized, they are inactive in nature. So what it does, it undergoes the glycosylation process. under which it adds the sugar residues on the nitrogen molecule of the protein and makes it activated. Now, once it gets activated, now it packs the protein into the vesicles and transports it to the Golgi apparatus for its further modification. Next organelle we have is smooth endoplasmic reticulum. So you can see the structure of the smooth endoplasmic reticulum. There is the absence of the ribosomes. Since ribosomes are absent, so the synthesis of the protein does not take place in this region. So this region is responsible for the production or the synthesis of the lipids or the cholesterol. And its function mainly differs from tissues to tissues, like on the ovaries or the testes or testes are responsible for the like there they are doing the production or the synthesis of the steroid hormones, whereas the ones that are present in Leo, like the CYP450 enzymes, they are doing or helping in the detoxification process or in the biotransformation of the different drugs or the toxins and the alcohol. Now, it is also responsible for the synthesis of the glucose 6-phosphate, the metabolism of the glucose 6-phosphate, means it's doing the conversion of the stored glycogen into the glucose. Whenever we require glucose, what it does the conversion of the stored glycogen into the glucose. Now, it is also responsible for storing the calcium into the sarcoplasmic reticulums. Now, all these things which are synthesized from the smooth endoplasmic reticulum gets packed into the vesicle and is being transferred to the Golgi apparatus. So the next organelle we have is Golgi apparatus, also called as the Golgi bodies. Now these Golgi bodies are responsible for the modification of the proteins. What they do, they have a receiving vesicles. Whatever the material is being synthesized from the rough endoplasmic reticulum and the smooth endoplasmic reticulum, it receives it by the receiving vesicles, and it does its modification by undergoing glycosylation process and the phosphorylation reaction. Here in the glycosylation reaction, they're adding sugar moiety on both the oxygen and the nitrogen molecule of the protein, whereas in the rough endoplasmic they were adding the sugar moiety only on the nitrogen moiety. After the modification is done, the Golgi apparatus packs the molecules or the vesicles and transports it to its desired location or the destination for its further functioning. Let's see the next organelle. Next we have is the mitochondria. So mitochondria is also called as the powerhouse or the power station of the cell because of its structure. Like you can see in the inner membrane, it has some foldings, extreme foldings, which gives a shelf-like structure, and this is called as the crest. Now, the number of crest is directly proportional to the number of mitochondrial cells. For example, in a straight muscle, since it's undergoing simultaneously of contraction and relaxation, it requires more energy, so more number of mitochondrial cell and the more number of crests is present. So a great number of ATP, that is adenosine triphosphate, is getting is getting synthesized. Whereas in the fat cells which do not require the more energy, there is a less number of crests is present. So less number of mitochondrial cells. Since less number of mitochondria is there, so less number of ATP is there. This adenosine triphosphate is being synthesized by two processes, that is oxidative phosphorylation from the electron transfer chain or by the substrate phosphorylation. Now this ATP which is being synthesized acts as an energy unit and takes part in the helps in the metabolic reactions like Krebs cycle, urea cycle, heme synthesis, gluconeogenesis or the ketoneogenesis. So the next organelle we have is ribosomes. Ribosomes are the complex of the rRNA and the proteins which are synthesized in the nucleolus region of the nucleus. So they are of mainly two types, that is the ones that are bounded and the free ribosomes. The ones that are bounded with the membrane are called as membrane bound ribosomes, like the ones we have studied on the rough endoplasmic reticulum, and the ones that are present in the cytosols are called as the free ribosomes. What they are doing, their main functioning is the synthesis of the proteins, means they are taking part in the translation process, that is from RNA to protein. And the ones that are present in the cytosol, they are holding the nucleus, they are holding the protein inside the cytosol. Next we have is the lysosomes. Lysosomes are also called as the stomach cell. They are doing the digestion process of the different macromolecules. You can see the structure of the lysosomes. It is having a diameter of about 250 nanometer to 500 nanometer and is having somewhat spherical structure. Now what these lysosomes are doing, they are doing the hydrolysis, or we can say the breakdown of the macromolecules with the help of the different enzymes like protease, nucleotides, etc. And they are doing the autolysis of the damaged cells. The cells which are damaged and not good for us, they are doing the autolysis of that damaged cells. And if any organelle is not functioning properly or is been damaged due to certain reason, it does the autophagy of that organelle. So next we have is the peroxisomes. So this peroxisome is mainly responsible for the breakdown of the peroxide into the water molecule and the oxygen molecules. Now this peroxisome is also responsible for the breakdown of the free radicals, which are formed due to the reactions of the mitochondria. With the help of different enzymes like catalase or oxidase, it does the breakdown of those free radicals, and it also does the metabolism of the alcohol and is also responsible for the oxidation of the fatty acids. Now these peroxisomes are synthesizing a special kind of lipids that is present in the white matter of the brain, called as the plasminogen and the cholesterol. Now smooth endoplasmic reticulum was also synthesizing the lipids, but the one that is present in the white matter is being synthesized by the peroxisomes, not by the smooth endoplasmic reticulum. So the next one we have is microfilaments. Microfilaments is a part of the cytoskeletons. Cytoskeleton is mainly responsible for providing the structure and the strength or the shape to the cell. They are of three types: microfilament, intermediate filament, and the microtubules. So microfilament is providing the strength or the edges to the cell, and it has a special kind of monomer or the protein called as the actin, which are of two types, that is filamentous actin and globular actin. So this actin, when gets combined with the myosin, it helps in the muscle contraction, also takes part in the cytokinesis of the mitosis process, phagocytosis of the WBCs, and deposis of the WBCs. That is, it controls the shape of the WBC cells. Next one we have is the intermediate filament. These are the most stable filaments of the cytoskeletons among the all three, that is microtubules, microfilaments, and the intermediate filament. They have the monomers present, different monomers present at the different sites, like the ones that are present in the neurons are called as the neurofilaments, and neurofilaments, or the ones that are present in the nerve cells are called as the neurofilaments, or the ones that are present in the muscle cells are called as the plasmins. The ones that are present in the epithelial cells are called as the keratin, and the ones in the fibroblast are called as the vimentin. So what they do, they act as an anchor, means they are anchoring or holding the cell to extracellular matrix, or cell to the organelles. So next we have is the microtubules. So these microtubules are providing or responsible for the structural or great structure of the cell. So their functions is they act as an intracellular transporter, which is an ATP dependent. Like what it does, they have a motor proteins of dynes and kinesins, which binds with the organelles, and they transport the materials inside the cell. Next, they take part in the cell divisions, Means it's very easy for microtubules to get formed and to get broken down. So they takes part in the mitosis or the meiosis process of the cell division, where the separation of the chromosome is taking place. Next, they are helping in the cell extension or the cell motility. Means what they are doing, they are forming the base for the cilia which is present in the respiratory tract, or they are also forming the base for the flagella. For example, the sperm cell. This flagella helps in the movement of the sperm cell. Next one we have is the cell membrane. Whatever the organelles we have discussed till now are being surrounded by the cell membrane. This membrane defines the boundary of a cell and it separates the cell from the outer environment, and it is also responsible for transporting the material which is required inside and which is required outside the cell, and it is also responsible for facilitating the electrical signaling between the cells. So the functions of the cell membrane is controlled or being affected by certain criteria, like the presence of the cholesterol. If the more amount of cholesterol is present, the fluidity of the cell membrane gets reduced, and if the less amount, means the vice versa happens, the less amount of cholesterol is present, the fluidity increases. And since it's responsible for transporting the materials inside and outside, so it requires a transporter. So here they have proteins which act as a transporter. The proteins are of two types, integral protein and the peripheral proteins. The integral proteins are sometimes glycosylated, means they have sugar residues present in them. They are present in the integral region, so they are called as the integral protein. The ones that are present on the peripheral region are called as the peripheral proteins. Now this cell membrane acts as a barrier, means like everything and anything just can't go inside and just can't go outside. They have to undergo certain process or the diffusions, like simple diffusion, vacillated diffusion and the vascular transport. Regarding these diffusions, we'll be discussing about them in the next part, that is the part 2. So up till this, now let's revise or recap in the summary of what we have discussed till now. We have discussed that membranes are made up of phospholipids and proteins. Yes, they are made of phospholipids and proteins, and they are acting as a boundary for the cells. The components, like the organelles, are surrounded by the cell membrane, and the functions of the cell is being compartmentalized into the different organelles. Now, the mitochondria is producing the energy, that is adenosine triphosphate, and more than one mitochondria can be present inside the cell depending upon its activity. The peroxisomes is doing the destruction of the hydrogen peroxide into the water and the oxygen molecule. Then endoplasmic reticulums are responsible for the production of the proteins. Golgi apparatus is mainly focusing on the modification of the proteins. Lysosomes are having the degradative enzymes which are helping the breakdown of the macromolecules. Then cytoskeletons are providing the strength and the structure to the cells, and they have microtubules, microfilament and the intermediate filaments. Then microtubules have alpha, beta and the GABA tubulins, and these microtubules they have the proteins alpha, beta, and one more is there, that is called as the gamma 1, but gamma 1 itself does not take part in these microtubules, but what it does, it's present in the centrosome, and it is responsible for the assembly of the microtubules. Next is the microfilament. It is composed of actins and is providing the strength to the cells. Then intermediate filament is the most stable one. So up to this, we have discussed about the cells and cell structure and its functions. So thank you. If you have any query or if you need notes on this topic of the part 1, that is structure and function of the cell, you can contact me on my Instagram ID, that is shine210998. Do subscribe my channel and follow me for the more updates. Thank you.